good morning to everyone. I am Bhadresh Rami. I head the protein sciences and the cell and genome engineering team in Discovery Biology unit in Syngene International. We have a dedicated team for performing gene editing and latest CRISPR technology approaches in our team. To come to your question, gene editing is a type of genetic engineering approach in which deoxyribonucleic acid, more popularly known as DNA, is inserted or deleted, modified, or specific fragments replaced in the genome of a living organism or living cell. Gene editing essentially is a targeted approach that helps to insert or delete at site-specific locations, unlike earlier, more historic gene engineering approaches, which were not very precise. Some of the commonly used gene editing techniques are either based on enzymes called as meganucleases or zinc finger nucleases, ZFNs, or another class of proteins called talons, which is transcription activator-like effector-based nucleases. And of course, the currently most popular approach called CRISPR, which is an acronym for clustered, regularly interspaced, short palindromic repeats. So together, all these approaches constitute kind of a kitty for performing gene editing studies. That's an excellent question. And to answer that question, we need to go a little bit into the history of the CRISPR-Cas technology, how it came about, and the beauty of how a very distantly related scientific discovery has led to what is today a Nobel Prize-worthy effort. So CRISPR, as we mentioned, is clustered, regularly interspaced, short palindromic repeat. It's actually a bacterial immune mechanism, which helps bacteria to, fight or combat or deal with invading bacteriophages. These are viruses that attack bacteria. And how the bacteria have evolved their own kind of primitive immune system using this CRISPR approach is that pieces of DNA from the viruses were nipped off by the bacteria. And in some sense, these sequences of the viral DNA provide a kind of a primordial memory for the bacteria to remember and recognize these threatening viruses in the subsequent infections on the bacteria. So the next time a bacteria senses a virus, they produce a customized RNA based on these stored viral DNA sequences. These RNAs help in production of an enzyme called CRISPR-associated enzyme, and there is various varieties of it. The most popular is the Cas9 enzyme. These enzymes are then used to chop off the invading viral DNA and thus destroy their genetic material, preventing them from continuing with their infection process. So CRISPR actually occurs in its natural form in bacteria as a defense mechanism, where these molecular scissors, if we may call them, the Cas enzymes, they recognize the DNA from viruses. And what the current Nobel laureates, Charpentier and Doudna, have demonstrated is that this mechanism could actually be precisely tailored and controlled to cut any DNA molecule at a predetermined site of our interest. Okay. And over the course of years and decades, the development of this technology with further refinement and precision has enabled today's scientists to modify DNA sequences in a wide range of either cells or even organisms. And it is used broadly in the fields of biotechnology and in the development of future therapeutics in the entire field of drug discovery. To give you a few examples, early this year, a person with hereditary blindness became the first individual to have a CRISPR-Cas9 based therapy directly injected into the person's body. And towards the middle of the year in June, it was announced that two patients with beta thalassemia and one with a sickle cell disease would no longer require the very painful blood transfusions after their bone marrow stem cells were edited using the CRISPR technology. So thus, the CRISPR-Cas9 system has provided scientists with a highly manipulable and, as of today, a relatively easy and effective method to edit genes, as per the requirement of the disease or the therapeutic area or the downstream application. And it has opened up a whole new universe where matters related to human health and disease can be addressed. So yes, it is definitely worthy of the Nobel Prize. Yeah, that's a very pertinent question in current times. Syngene, especially our discovery biology department and my team, has over the years built a very strong and consistently improving and refining our gene editing capabilities with expertise in CRISPR-Cas9 approaches. I'm proud to say I have an extremely capable and experienced cell engineering and genome engineering team here who are supporting several clients with some truly stellar and cutting edge work. To give you a few examples, we can currently actually knock out and knock in genes of interest for the clients in various different cancer cell line lineages. In that process, we have developed knockout and knock-in cell lines using the CRISPR-based approach in epithelial cell lines, blood cell lines, endothelial cell lines, and a variety of cancer cell lines. So if you are interested in this work, for more information on our gene editing capabilities, please do get in touch with us. Thank you so much for your time today, and I do look forward to hearing from you soon. Thanks.